Okay, so let's start by taking a look at genomic libraries. The first thing you might wonder is, what is a genomic library? So a genomic library stores the genome of an organism inside of cells. So genomic libraries are living collections of the genome. And the way that, the overview of how you would make a genomic library is you would take the genome that you want to keep in this library, cut it up into fragments, and then you insert those fragments into cells that you can maintain in the laboratory, and then you can either keep those cells going in culture, or you can freeze them for a later use. You can freeze them for storage. Okay, so the genome is pieces of DNA, so you can't keep them on a shelf, right? You keep them inside of cells, and so when you make a genomic library, you're storing that genome inside of cells. So if we take an overview picture, here's some cells, and we're after this genome. So we're going to extract the DNA from these cells, cut it up into fragments, and then we need a way to get those fragments into cells. So you're going to put them into what's called a vector. And a vector is something that can introduce DNA into cells. And one vector that we're using in our laboratory is plasmids. So you can take this DNA fragment, you can put it into a plasmid, and then you can get bacterial cells to take up that plasmid, and then what you end up with is a collection of bacterial cells, each of which contains a fragment of the genome from the original cell, right? So this could be a human cell, and these could be bits of human DNA, and they could be in these plasmids, and you can see each bacterium now has a scrap of human DNA. So we are storing that that original genome inside of these cells. In order to cut DNA into fragments, one of the tools that's frequently used are restriction enzymes. Restriction enzymes, here's an example of one called EcoR1. They recognize a certain sequence in the DNA, and then they cut at that sequence. And this dashed line shows how EcoR1 cuts DNA. And this would be the sequence, which is called a restriction site, that EcoR1 recognizes. So if this is a piece of DNA, so a double helix, this purple and pink, and in the middle here we're seeing the sequence revealed, anywhere along this DNA that this sequence exists, EcoR1 would bind and cut, and you can see it makes this little asymmetrical cut, and then that would separate this piece of DNA into two fragments. And when restriction enzymes cut, frequently they create what are called sticky ends. They cut basically at a palindrome, a mirror sequence. See this AATT, TTAA? And so when it's cut at this asymmetrical kind of mirrored sequence, there's a tendency for these sticky ends to come back together and bind to each other with hydrogen bonds. That's why they're called sticky, because they want to come back together. And that becomes useful when you want to insert DNA into other DNA, and we'll see that in a minute. Okay? So if I was going to make a genomic library, one way that I could do that is I could extract the genome from the cell, the genome that I want to store. I could cut the DNA with restriction enzymes and get these sticky ends. All right, so if we were to take a look at what we would do then, I could take those pieces of my genome that I've cut, and I have a plasmid that I want to use as my vector. In this figure, we're using a plasmid. So here's this plasmid, and I would cut the plasmid with the same restriction enzyme that I used to cut my DNA. So the DNA that I want to store is called my gene of interest, right? So I've cut that with EcoR1, let's say. I cut the plasmid with EcoR1. That opens up the plasmid. And because I've used the same restriction enzyme on my DNA fragments and on my plasmids, they have the same sticky ends. So if I mix them all together now, there's going to be a tendency for those sticky ends to come together. Sometimes the plasmid might just close up with no fragment because it'll stick to its own sticky ends. But sometimes those little fragments of DNA are going to stick to my plasmid and get in there and be introduced into the plasmid. So I'm going to create a population of plasmids, some of which have the fragments from my genome and some of which don't. Okay? And so this is just showing that again, showing these little fragments sort of inserting into these plasmids. And remember that the sticky ends are only bound to each other by the hydrogen bonds between the bases. Now, if I want to seal those DNA fragments into those plasmids so they don't separate out again, I need to seal up the breaks in the sugar phosphate backbone. And you remember from DNA replication, an enzyme that does that is DNA ligase. So I'm going to add some ligase to my mixture of plasmids, and then ligase will come along and seal up these breaks. So now my fragments will be sealed into the plasmids. And once I've done that, then I can mix my plasmids with my bacteria that are going to be the host cells for my library. And I can do a procedure like you're doing in lab this week. I can do a little heat shock and get those cells to take up those plasmids and be transformed. So I'm going to transform the bacteria with the plasmids. And some of the bacteria will take up plasmids, but not all. All right, so to clone my genes, I need a vector. And that's going to be, a vector is something that can carry DNA into a cell. Plasmids are commonly used. Plasmids can handle small pieces of DNA. There are other things that can handle larger pieces of DNA. There's something called a bacterial artificial chromosome that's sometimes used, or sometimes there's a yeast artificial chromosome, and yeast are eukaryotes, so I can store a genome in yeast cells too. And as I said, I'm going to cut my vector and my DNA to be cloned with the same restriction enzyme so they have the same sticky ends. Mix them together. Some vectors will pick up DNA fragments, and that'll be through the hydrogen bonds. And so then I'm going to add DNA ligase to seal up those breaks. Okay. Then I transform the bacteria with the vector. Now I have two issues. One is that I have some bacteria have picked up the vector, the plasmid, and some haven't. So I have to deal with that fact that some of my bacteria have the plasmid, some of them don't. The other thing I have to deal with is some of those plasmids have a piece of my DNA that I wanted to clone, and some of them don't. So right now I have kind of a mixed bag of things I want and things I don't want. And I've got to clean it up so that all I have left are my bacterial cells that have the vectors and they have the pieces of the genome. Okay, so there's two things I'm going to do. I'm going to select for cells that have the vector, and I'm going to have to check somehow that their vector has the DNA fragment. So the way that I can do that is when I choose my vector, I don't just pick any plasmid. I either choose a plasmid that has certain features, or really more commonly, scientists will build a vector, and then you can just buy them from a supply company. And a good vector is going to have two things. Well, it's going to have more than two things, but two things that relate to what I'm saying. One is it's going to have a gene for antibiotic resistance on it. That's going to allow me to kill off any bacteria that didn't take up the vector at all. So I want that. The other thing it's going to have is what's called a reporter gene. It's going to have a gene in it that somehow is going to signal to me whether or not the vector has taken up a fragment of DNA. And so in this example, in this picture, they've included in this vector the LAC-Z gene, which is the gene for beta-galactosidase, right, that we talked about when we talked about the lac operon. And there is a molecule that when you put it with beta-galactosidase, the beta-galactosidase will break the molecule, and so the molecule itself is blue. No, how does it go? No, sorry, I got it wrong. When the molecule is broken by beta-galactosidase, it turns blue, right? So in the presence of beta-galactosidase, you get a blue color, okay? And then what you do with your plasmid is you have this gene, this reporter gene, this beta-galactosidase gene, and so if this gene is intact, if it's in the plasmid and it's working and you get the enzyme, you'll get the blue color. But there's a restriction site in the middle of the gene. And so when I cut the plasmid with my restriction enzyme, that gene gets cut, right? And if this plasmid picks up a piece of DNA fragment, that gene is never going to be whole again. If you cut this circle with a pair of scissors and open it up into an open ring, and then you slip something in there into that open ring, the gene, which is red in this illustration, is going to be separated by the DNA fragment. So any vector that has picked up a DNA fragment is no longer going to have a functional beta-galactosidase gene. So those bacteria, when they're exposed to that molecule that turns blue, they're not going to turn blue because they can't make the beta-galactosidase. They can't make the blue color reaction. Okay, so I have two things that are built into this plasmid that I need. One is the antibiotic resistance gene, and the other is the reporter gene that's going to give me some kind of a color change that tells me whether or not the fragment has been picked up by the plasmid. And so after I transform the bacteria, right, first thing I'm going to do is I'm going to hit that mixed bag of bacteria. I'm going to hit them with my antibiotic. Any bacterium that actually picked up a plasmid is going to survive the antibiotic because they have the antibiotic resistance gene. Any bacteria that didn't pick up the plasmid are going to die. And that's good because I'm not interested in them, right? I only want a collection of cells that has picked up my vector, has picked up the plasmid. But notice in this illustration that some of these vectors, like this one that has this continuous pink band here, they don't have a piece of DNA that they picked up in the vector. They don't have a scrap of my genome, right? Some of them do. So that's when I come to the next part, which is I'm going to look at my plates. I'm going to put these bacteria on plates, and I'm going to look at what color the colonies are. If the colonies are blue, then I know that that reporter gene was not interrupted, and they can still make beta-galactosidase. The colonies are blue, so they have a vector, they have a plasmid, but they're not useful to me because they don't have a piece of DNA inserted into the plasmid. Any colonies that are colorless, that tells me they did pick up a scrap of DNA in their vector because they can no longer make the enzyme and they're colorless. Okay? So I've got resistance genes in my vector. That's going to let me select for transformants, right? And when we used antibiotics to select for resistant bacteria, we're using positive or direct selection. We're going to kill off any cells that don't have a plasmid. I also have a reporter gene that allows me to select for the transformants that do have a piece of the DNA of interest, that do have a piece of the genome. So my vector has a reporter gene, example, a gene for an enzyme that makes a color change, and the reporter gene has a restriction site in it, it has a cut site, and if there's a piece of DNA inserted into that reporter gene, it's going to be a mutation, a knockout, an interruption of the reporter gene so that I'm not going to get the color change. Okay? So once I've done all that and I've got my colonies growing on plates, right, all I have to do is look and pick up the colonies that I'm interested in, so the colorless colonies that I know have the vector, have a piece of DNA, and I can take a little sample from each colony, put them in different tubes of media, and I can have a whole, racks of test tubes, each that has a different colony, so it has a different clone, we call it a clone, from my library. And each of those clones has a fragment of DNA. I don't know what's on that fragment of DNA. I don't know if some clones have a similar piece of DNA. At this point, I don't know. All I know is I have a collection of cultures or clones, each of which has a fragment of the original genome. That is my genomic library, that collection of clones. Okay, so I pick my colonies, and each one of these clones, I culture them. Okay, and then I can maintain them in culture, or I can freeze them for future use. Okay, so that's the end of genomic libraries. Next, we're going to take a look at gel electrophoresis and PCR, both of which are also important